Welcome to Bioprospecting 101. Bioprospecting is the search for plant and animal species from which medicinal drugs and other commercially valuable compounds can be obtained. For an example, take this pit viper found commonly in Borneo. This is a Bornean-keeled green pit viper, and although it is venomous, the venom can be used to save lives. Venom is primarily used to subdue its prey quickly, but it is also used for self-defense of the snake. If bitten by the snake, there is a possibility of severe tissue necrosis taking place at the bitten area. Venom of the snake is cytotoxic, which means it has cell-destroying capabilities. Venom is composed of a cocktail of proteins. Every type of protein present in the venom has its own role to play. Do not attempt to handle wild snakes on your own. Wild snakes are unpredictable and dangerous. Snakes should only be handled by trained professionals. The venom was extracted from the snake and the footage was not included for safety reasons. As you will see in a while, texture of venom is slightly viscous and usually slightly yellow in color. Venom is produced only in smaller quantities due to its potency. Once the venom is extracted, separation techniques will isolate the desired proteins from the venom in the laboratory. Cancer cell cultures are grown within the laboratory. The extracted proteins are then introduced to the cell cultures to determine the effectiveness of the compound. Scientists then check the affected cells for the mechanism of action at a molecular level. This is to determine if the compound is suitable to proceed with the next stage of the research. Next stage of the research is to perform preclinical trials. Intensive tests will be carried out on lab rats and primates for a longer period of time to make sure the compound only destroys cancer cells. Once the preclinical tests show positive results, clinical testing will begin with voluntary patients. This will make sure the product is stable and safe.